we have to ask ourselves, Tri-Creeks, is it relevant? What did it teach us? Did it tell us anything? And is it relevant to today? There was a very different mindset in 1960 in terms of logging, and it's different than today. So a brief outline. I've got about 28 slides, and I'm going to try to get through them all. I tend to ramble a little bit, so I'm going to try to stay on track, and perhaps we'll have a little bit of time at the end for some questions. A little bit on the origin, objectives, and the design. Some of you may not know where Tri-Creeks is. A little bit about the fish community and the results. Did they teach us anything? And discussion and data gaps. Are the results useful? How might they fit into today? And I have to say, wow, I mean, this is really stepping back, right? I cut my teeth here. We didn't have technology. I used a little Texas Instrument, wrote my programs on a little magnetic strip. I'd get 100 N into my sample, and I'd make a mistake and have to start over again. And today it's amazing. So the origins, conceptualized in 1960 at a natural resources conference here in Edmonton, perhaps like this. Maybe this is the beginnings of something really new, who knows. And then in 1965, the department at that time, under the IHD program, which started in 1965, developed the Alberta Watershed program, and Tri-Creeks was spawned, for lack of a, you have to pardon the fish pun. And partners. At that time, the major partner was St. Regis, or did Northwestern Pulp and Power predate St. Regis?
Yeah.
Who said yes?
It did,
Yeah, it did. Okay. And in 1965, we had access constructed into Deer Lake and Eunice. There was an old access road into the Wampus Basin, but we needed access into Deer Lake and Eunice. Gauging took place, Streamflow, groundwater, stream temperature, precipitation stations. A field camp was brought in, and we closed the creeks to fishing, and we began. And these were our objectives. And perhaps some of these, if we reword them a little bit, might still be applicable today. Back then it was clear cutting, and of specific interest was how it might influence the survival, growth, and reproduction of fish and aquatic inverts. And specifically, what would the effects be of removing streamside riparian vegetation on water temperature, stream flow, and suspended sediment? And then how does clear cutting affect the hydrological regime? And what are the effects of road construction and clear cutting on suspended sediment and channel siltation? And then the last one was the hydrochemical regime, and to me it's just water quality. And the design. Today we probably wouldn't do a watershed study in this fashion. We used a paired basin approach back in the mid-60s. This was sort of the standard approach to watershed studies. In this particular case, required three similar streams. One to be a control, one to be experimentally manipulated, the other one would be standard cut, and we'd see what the results would be. And planned as a 20-year study, and the title says 30 years of fish and forestry research. Well, it was planned as a 20-year study, so the door closed 1985. and it did. It got slammed shut. We managed to carry the project for 2 more years because we had some flood events in the watershed, and we lost a couple years of data. But 1987, the door was slammed shut. So where are we? If you don't know where Tri-Creeks is, south of Hinton, about 40 clicks, Wampus, Deer Lake, and Eunice, about 60 square kilometers. Maximum elevation is about 1,700 meters above sea level. It's a typical upper foothills eco subregion. And I'm not going to go beyond that. Fish, that's my real interest. We've got native rainbow trout and bull trout, and we're now calling these invaders, but we brought them here. We put them everywhere, brook trout, and we have some issues. These 3 fish predominate the fish community in these small 4th order streams. Rainbows tend to be really abundant. Subadult bulls are present foraging, and we get annual migrations into some streams, to very specific streams, in fact, for spawning by bull trout. Brook trout are at the same trophic level, and so as rainbow trout, and so they compete for the same space and food, and I'll talk about that a little bit more as we get into the presentation. There are other species present: mountain whitefish, white suckers, long-nosed suckers, long-nosed days, burbot. Some streams have all of them, some have more, some have fewer. It's quite variable, can be quite variable. And so I'm just going to jump into the results, and I'm not going to spend a lot of time here because I think some of this, if you really want the results and you want a little more than what I can give you here in the time that I have, you need to dig up some of these reports, and most of them are available in the GOA library. They're all there. Some of them have ISBN numbers. I was really surprised. Somebody says, hey, you should Google your name. You know, some of your old Tri-Creeks reports pop up, and they're in the environment libraries where they are. This is just going to be the Coles notes for some of this stuff. Geology and soils, highly erodible in 20% of the study area. So we got high probability for erosion. Climate, I couldn't believe this when I read it. Mean annual temperature is only 1 C. No wonder I froze out there half the time. Cool, wet summers, long, cold winters with occasional warm Chinooks, and it really is that. I mean, it's barely a 6-month open water season. And it's not very much precipitation. 885 millimeters, and 36% of it falls as snow. Timber harvest. So 1965 to 1975 was supposed to be the calibration, pre-harvest or calibration. Road construction took 3 years as opposed to the 1 year originally planned. And then the harvest treatment, we had winter logging in both basins, but originally planned to happen in 1 year, happened 2 years in one basin and 3 years in another. Cut block sizes were quite variable, 20 to 114 hectares in Deer Lake, up to 200 hectares in Wampus. And at that time, 4 of the 34 cut blocks actually exceeded the ground rules at that time. They harvested 50% of the merchantable timber in both watersheds and about 40% of both watersheds. And post-harvesting monitoring, I mean, this became one of the big sticking points. There really wasn't much of a post-logging evaluation. So Streamflow, what did we see? A marginal increase in Wampus Creek, and this is seasonal water yield because the gauging stations weren't really set up to deal with winter flow. Icing conditions were really severe, and so it's really open water seasonal water yield, 7% increase in wampus, 22% increase in deer lick. And we had buffer removal in deer lick. You know, do the two connect? Both increases, or the increases in both basins, neither of them were deemed to be significant. And a little bit more of that later in terms of how it might relate to 22% increase in water yield might not be significant, but it might have a biological significance, and we'll talk about that a little bit more. Water temperature. Well, not surprisingly, the mean water temperature didn't increase following logging, but the monthly maximums changed fairly dramatically. And as expected, although there was no pre-modeling on any of this, the maximum water temperatures in deer lick increased the most, and that's where we had buffer removal. Remove the canopy, increase solar radiation, we expect to get warmer water. And we saw temperatures of 21 degrees Celsius, maximum temperatures of 21 Celsius, and we're beginning to approach the maximum upper limit for rainbow trout. 20 C, the literature says that 23 degrees C is, or can be lethal. It depends a little bit on acclimation. There are other variables. We didn't see any fish kills. We were close, but there were lots of thermal refugia for the fish to deal with during the hottest parts of the summer months. Suspended sediments, I have to apologize, the light blue doesn't really show up. And the pictures in the background are actually photographs from Tri-Creeks, the logging. This is, and you can't really see it, but it's a horrible haul road in Upper Wampus. Suspended sediments, highly variable, and there were significant increases in both the Wampus and the Deer Lake subbasins. In Deer Lake, we saw actually much higher increases, particularly the 2 years following road construction. The first year there was just road construction, 26% increase in suspended sediments, and the second year following the construction of the road, a 52% increase. And then following logging, there was another, suspended sediments were 19% higher than predicted. And the calculated yield ratios suggested that Wampus Creek produced less sediment than Deer Lick, and of course Deer Lick was unbuffered and Wampus was buffered. So, nutrients. There's a lot of water quality data got pitched because there were quality control issues, but from the data they were able to analyze, and I don't know why some of these anomalies occurred. I'm not a water chemist, and so I don't understand what was going on here. But in Eunice Creek, which was a control, we would expect that there would be no changes, and yet in terms of inorganic nitrogen, there were significant increases following logging. Why that occurred, I don't know. Total phosphate loading did increase significantly in both Deer Lick and Wampus following logging. And the graphic behind the slide is actually the visual impact that the nutrient loading had on Wampus Creek. And this persisted for about 3 years during the post-logging phase. And once we had the nutrient flush through the system, the creeks returned to normal. And we do see algae blooms like this now, but they're not as extensive. And this had a significant influence on fish growth. And I'll get to that in a minute. And this significant increase or decreases in pH, like 0.1 to 0.2 decrease, it was significant, but biologically wasn't really significant. So the fish community. So I just put up this quick graphic because there are So we have fish data from 1969 all the way out to 2011 here. The study ended right here in 1986. And like I say, the door slammed shut. And we managed to get back. Some of us decided we were just going to go on a camping trip. We took a backpack shocker with us and we went back in 1990 and we collected a little bit of data just to see what was going on. We couldn't let this sleep, right? And I mean, we had an ADM in 1983 actually here tried to close the project in 1983. In fact, he did. He slammed the door and we went on to do other things. And we had some folks at in Hinton who didn't like the idea that the government was going to pull a pin on their watershed study. And we were back in business in September. And so we managed to see it through to its projected life. But then the door slammed shut, and most of the reporting that occurred in the mid to late 80s was all piecemealed. A few funds were found here and there to help folks with write-up and stuff like that. And then the model forest came online, and through that program, we were able to begin again to look at the fish community, and in particular rainbow trout densities, and we've maintained that ever since. And you can see that populations bebopped around. This decline, this is a flood, a 1 in 100-year flood. 1980, this is a 1 in 100-year flood. There was high water in here, and this is really the post-logging phase, and we weren't there. We missed all of that. We don't know what the responses really were in terms of the fish community. But by the time we get down to here, 10, 15, now 25 years after the study, things have started to change. I mean, we get same kind of densities in lower Wampus. We're getting a shift in the fish community. Has this been facilitated by what we've seen change within the stream itself in terms of habitat parameters? Upper Wampus, I mean, these last here in the early, this is the early 2000s. I mean, we had densities that we'd never seen before, and this is after logging. Lower Deer Lake, same kind of thing. Increase in brook trout in lower reaches. Upper Deer Lake, pardon me, we're seeing some increases, but we're in this case. I mean, we're 25 years down the road from when the study concluded. And then we get to Eunice Creek, and I haven't put Upper Eunice on because it's pretty harsh climate. We don't have very much data up there. But in Lower Eunice, we did have, these yellow bars are actually bull trout, and every year we would see half a dozen to a dozen individuals enter Eunice Creek to spawn in the fall. And some years we had more, some years we had less. And we're missing a bunch of data. We went back in early 2000. Bull trout are still there, probably somewhere around the same. Last few years we've been going back, and we're not finding any fish. There's just no fish anywhere in Eunice Creek. No brookies, no bulls, no rainbows, and we don't know what's going on. Eunice was the control, and wasn't logged until we got to the early 90s, and then it actually there was some harvesting occurred in Eunice Creek. How that relates to what we see in the fish community, I have no way of knowing. There's no way. So trying to integrate some of these things, the physical changes that we saw with fish, trying to make some sense out of what it might tell us. Well, Streamflow was the only variable that we could find had a relationship with fish, and specifically recruitment success. And so this is just total water yield during the incubation period, not annual yield, but incubation period. And this is rainbow trout fry density. And this yield, I have to say too, is the yield that's above this threshold line. So in Wampus Creek, we had the good folks from Alberta Environmental Water Survey section come out, and we spent a week or two painting a bunch of colored rocks, and we put them in the stream channels. The rocks were the same particle size distribution of spawning substrates, and we painted them, and we put them in spawning habitat. When the water flows got higher, we went and looked how far downstream these painted rocks got. We listened on hydrophones, and we calculated this threshold. 0.7 cubic meters per second, the mean particle size began, of spawning substrates began to move. We computed the yield from the third week of May to about the 10th of August. That was the incubation period. Computed the yield above this threshold and plotted that against fry, and we have a relationship. Temperature and fish, well, we spoke a little bit about temperature before, but we really, there were no correlations with any of the temperature data and fish population. I mean, there's an obvious relationship between thermal units and incubation period, and it works out to about, I think it's 590 degree days. So at a mean temperature of 10 Celsius, it takes 59 days for the eggs from when they go in the gravel to when they pop out of the gravel. So we have that relationship, and it's a very tight relationship. But in terms of the increases in water temperature and the actual fish densities, there was no relationship. And so how might temperature increases influence the incubation period was never followed up on. We never did any modeling to see whether elevated water temperatures would initiate earlier spawning and earlier emergence, and how that might impact population sustainability. And this last point, I've already mentioned something about survivability in terms of high water temperatures. So settled sediments, suspended sediments were so variable, it was really hard to get a handle on with respect to fish. But we know that settled sediments have a dramatic impact on spawning for rainbows, for salmonids in general. And so we looked at that relationship, and percent fines is a parameter normally used or generally used, but it's not very descriptive. I use the Friedel index down there. The Friedel index just speaks to permeability and pore size, and it's a better descriptor of the quality of spawning substrate. But there is a relationship between percent fines and the Friedel index, and experimentally we showed that an increase of 10 to 20%, that's 100% increase, right? Results in a fairly dramatic reduction in fry escapement. So we were able to show this at Tri-Creeks, but we couldn't relate changes in the population size to sediments in the stream, because the literature tended to said that you needed to get above 20% fines to adversely affect populations. And we never really achieved that. In spite of what went on there for logging, we never really got above that high sediment load. I mean, when you plot the data, the sites that had really high sediment didn't produce any fry, but generally most of the sites didn't have that level of sedimentation. And the interesting thing is that after, during the post-logging phase, the Friedel index, so the quality of the spawning substrates, actually increased during the post-logging phase. And that was because of the flood in 1980, which happened right between the pre and the post-treatment. And floods are renewal for stream channels. They scour and remove fine sediments. Fire renews the terrestrial landscape. Floods renew spawning substrates. Very dramatic. They decimated the populations in 1980, and within 5 years, we had populations, if you remember the earlier graphic, within 5 years, populations recovered to pre-logging densities. So the flood really masked this effect at Tri-Creeks. So nutrients, the algae blooms, male salmonids generally grow faster than females. So the lower line here are female rainbow trout. The middle line here are male rainbow trout. You look at just about any salmonids, whether it's bull trout, cutthroat, Chinook salmon, coho, males tend to be larger than females. Not true for pike. You catch a big pike, it's always a big female, right? But not for salmonids. And they don't grow very much. I mean, this is 10 years of age. That's the oldest fish we ever saw. They get about 20 centimeters. So these are mature fish living in these little creeks, pretty small. And we saw a significant increase in growth because of this nutrient flush in the first 3 year classes following logging. I projected the line. We don't know if this growth for these fish, the green triangles here, would have persisted because we never ever went back. And by the time we did go back, these fish were already out. We were into the next generation, and so they were gone. But for the 3 years following logging and during the period of this nutrient flush, those algae beds were just packed with young of year rainbow trout. And they were growing like gangbusters. They were almost twice the size than the pre-logging fish. So a fairly dramatic effect. So I'm going to try to wrap this up here. Project concluded in 1986, and we talked about the write-up was piecemeal. Some of it is still just now being written up. We've pulled the file drawer out, and we're looking at all the winter habitat data that we collected, trying to classify critical wintering habitat. These fish winter in these little streams. Before freeze up, we have about 5 years of 5 winters of data, about 15 pools per winter. The mean maximum depth for wintering pools is, I think it was, 0.65 meters. And by this time in the winter, the volume under the ice has been reduced by about 80%, the mean volume. So they're living in very little water at this time of year. And we're just writing that up now. Changes were observed in streamflow, sediment, temperature, and nutrients. And fish changes were not well linked to those changes. And the fish habitat changes were largely unmeasured. Rich did some work down there and showed us that there were clearly some channel changes. But a lot of the stuff that I saw as channel changes because of the removal of riparian vegetation in Deer Lake, for example, is just anecdotal. 15 years after logging ceased, you could walk and see the banks breaking in and the fish habitat changing. And now 25 years after, that same chunk of creek has some pretty high densities of rainbow trout. So habitat has changed, and we have to ask ourselves, well, what has the effect really been on fish densities? The experimental design has been criticized because of the paired basin approach. Those results are basically equate to that chunk of landscape. How much of those results can we project to within the same subregion? Probably very little, because at Tri-Creeks, for one thing, that hasn't changed is 1980 to 83, we had an initial disturbance, and there's been no other activity basically since then. Every other chunk of real estate in West Central Alberta has got oil and gas on it, it's got a coal mine on it, we've got all kinds of things happening. And the original Tri-Creeks study area, there's been some drilling. They've been looking for gas. They still haven't found any. There's a couple of, I think, minor producers of gas. And that's it. There's some potting going on, but other than that, there's really nothing else going on. Okay, some interesting stuff, and I'm just going to, where to now? So we're not going to log the buffer strips like we logged in 1980 in Deer Lake. I mean, that's, I think, a given. We're not going to go down that road. But if we do go into a new concept of riparian management, what will it mean? I just pulled the next two graphics out of a recent journal of fisheries, American Fisheries Society Fisheries Journal, and they're actually mapping water temperature and changes. So this is, and you can't read this, so this one here is rainbow trout, and this one is bull trout, and they've used spatial analysis and temperature modeling to predict changes in habitat suitability. And the big patches are fire. They put it on the landscape. And this is 1993 to 1996. So these are the time frame. Black, and you can't see it, there are channels that are marked. These are losses. Red is unchanged habitat. There are some green channels. You can't see it, sorry. The next one is sort of along the same lines, and I think this is maybe where we need to go with respect to any kind of riparian management that we embrace. We need to model temperature effects on rainbow trout and how encroachment by invasives might be affecting them in the long term. And so here again we have, use the pointer, this is 1978, B is 1970 or 2007, and this is the white are where temperature changes have changed. So this is blue is less than 9C, white is greater than 9C, and you can see that in 2007 there's more stream channel that's warmer, and so this is looking at the habitat loss for cutthroat trout and how it might facilitate the invasion of rainbow trout. And again here, this is 1978, and there's the red, there's an area here that's shaded in as red. So in 1978, very little of this watershed was suitable for rainbow trout, but in 2007, using temperature modeling, they've quantified where rainbows could invade and potentially hybridize with cutthroat, and so evaluate risk. And I think that's where we need to go. Quickly, we need to better understand this relationship. Flow increases during the incubation period rather than annual yield, and some of you have heard me say this before. Complete removal of buffers, I mean, that's not supported, but what effect does forest harvesting at some scale in riparian zones have on these particular parameters? And my conclusions. And can we model, and I think there are lots of tools, can we model the effects of riparian logging to help understand risk to fish populations? And for Athabasca rainbows, that will be fairly critical. They're uniquely adapted to the upper parts of the Athabasca watershed, and many populations are isolated. And what risk are those populations at of potential extirpation? Okay, and I'm going to end there, and I don't think we have time for questions. And this is Upper Deer Lake Creek with the riparian buffers removed, and there are lots of fish there now, 25 years later. The pine stand here now is nearly 3 meters. So there's been a fair bit of regeneration. Okay.